Hi, I'm Angeles Aroca from Bassham Lab in Iowa State University, and I'm going to talk about persulfidation and the role of sulfide in selective autophagy under ER stress by the regulation of ATG18a. As you know, sulfide is a well-known poisonous gas that can be harmful for animals from very low concentrations in prolonged exposures to even death in severe cases. In plants, sulfide has also been always considered a toxic soil compound since it reduces nutrient uptake by reducing root respiration. Nevertheless, in the last decade, sulfide has emerged as a new signaling molecule as important as other transmitters in mammals, and also as important as a hydrogen peroxide and nitric oxide in plants. Almost 10 years ago, this publication was released demonstrating that sulfide was a signaling molecule and its way of action was through a post-translational modification of proteins that was originally named as sulfidation and later changed for persulfidation. But nowadays, you can still hear both names in papers. This modification affects the cysteine group of a protein, modifying the thiol group into a persulfide group. In this publication, they also describe a method to identify modified proteins, which was based in a previous method to identify a similar modification that is called nitrosylation. Since then, several new improved methods to identify persulfidated proteins have been published, most of them based in a similar technique. First, the thiol group must be blocked by a specific reactive, and then the disulfide adduct can be labeled with another specific reactive that usually attacks this disulfide bond. Using one of those methods, named tag switch method, we were able to identify more than 3,000 proteins in Arabidopsis plants, but about 2,000 proteins in all biological replicas. According to Mapman classification, these proteins were involved in a huge variety of biological processes like RNA or DNA binding, transport, signal transduction, response to abiotic and biotic stimulus, etc. Among these 2,000 identified proteins, there were some proteins that belonged to the core machinery of autophagy. So these proteins, these ATG proteins, were also targets for persulfidation. And some of you would say, what's autophagy? Well, it's a conserved regulated mechanism of the cell that serves to remove unnecessary or dysfunctional components. The unwanted organelle or aggregated proteins are enclosed into a double membrane vesicles called autophagosomes, which are then fused with the lysosomes or vacuoles to degrade the contents. This degradation process is critical to maintain cellular homeostasis, ridding the cell of either excess or damaged organelles, aggregated proteins, or even pathogens. But this process needs to be very tightly regulated because too much or too less recycling, too much or too less autophagy could cause serious problems to the cell and the organism. There are previous works from Dr. Gotor and Dr. Romero that demonstrate that sulfide is able to regulate negatively bulk autophagy induced by nitrogen starvation or carbon starvation. And also, some years ago, Professor Bassham from ISU published that ATG18a was involved in the ER degradation by autophagy under ER stress. These results drove me to think about the role of persulfidation of this protein in reticulophagy, which is the selective degradation of ER by autophagy. So what I asked myself was, could sulfide be involved in reticulophagy through the regulation of ATG18a by persulfidation? So the first thing that we did, we wanted to study the cysteine residue that was modified by this modification, persulfidation. And to do so, we expressed and purified the recombinant GST ATG18a protein, and it was analyzed by mass spec to search the mass shift for persulfidation in any of those cysteine residues. It was found that only one peptide, this one here in this graph, and here highlighted in red, was modified by persulfidation. Then we checked if this persulfidation was sulfide dose dependent, and to fulfill this objective, we used a new method that was published recently here, named pre-click labeling, which mainly labels cysteine residues with this fluorescent compound, that is chloronitrobenzofuran, and then only those adducts that contain this disulfide bond were able to be labeled by a dimidone-based fluorescent probe C5 with C5. After this labeling process, proteins were loaded in an acrylamide gel and scanned in a Typhoon fluorescence imager. This is how this scan looked like, and the line displayed in fire color shows the protein label with the dimidone-based C5, which means is the persulfidated fraction of ATG18a, while the line displayed in green is the fraction labeled with chloronitrobenzofuran, which serves as a loading control. In the graphs, the percentage of normalized band intensity was quantified with using ImageJ software, and is represented here, this intensity in the graph. So recombinant protein was treated with increasing concentrations of two different sulfide donors, and also with two different reducing agents, DTT and TSEP. And as you can see, increasing sulfide concentrations showed increasing band intensity in both cases, and a decrease in band intensity was observed in those samples treated with reducing agents. So we could say that persulfidation of ATG18a was sulfide dose dependent and reversible by reducing agents. The next question to be answered was to know if sulfide could regulate autophagy when it is induced by ER stress, which had been previously studied in mammals but not in plants. To demonstrate so, we used transgenic GFP-ATG8 plants. This protein, ATG8 protein, is a core autophagy protein that remains associated with the autophagosome. Here you can see it represented in this skin as a green spot, and due to it remains associated with the autophagosome is the reason why it's a key autophagy-related marker protein. 1 week old Arabidopsis seedlings were treated with tunicamycin for 6 hours to induce ER stress, and with sulfide for the last hour. Besides, to compare the effect of sulfide under ER stress was due to a ROS scavenging, other plants were treated with ascorbate and glutathione as antioxidant compounds. The results show that treatment with tunicamycin increased the number of autophagosomes, here the green spots, tiny spots, while treatment with only sulfide and the antioxidant compounds didn't have any effect reducing these autophagosomes. But treatments with tunicamycin and sulfide showed a decrease in the number of autophagosomes, while this decrease in the number of autophagosomes was not observed in those plants treated with tunicamycin and the antioxidant compounds. So we can say that sulfide regulates autophagy under ER stress, and that regulation is not due to aerobic scavenging. Furthermore, we wanted to investigate what happened with ER per se when it was stressed by tunicamycin, and what was the role of sulfide in this regard. To study this, we used plants transformed with GFP fused with the ER marker HDEL, these plants, and we analyzed the subcellular localization of the ER marker in response to ER stress induced by tunicamycin and sulfide treatment. Plants were also treated with concanamycin to prevent vacuolar degradation. The results show that without concanamycin, this line here, these pictures, no real differences were observed among treatments, and only typical ER structures were observed, like these ones here. Nevertheless, with concanamycin, an increase of ER fragments were observed in the vacuole in the plants treated with tunicamycin, which means there is an increase in ER degradation by autophagy. However, those plants treated with tunicamycin plus sulfide didn't show that increase in ER degradation. Therefore, we could say that sulfide regulates the ER degradation by autophagy under ER stress. Well, I haven't said yet that ATG18a is a protein of the protein family that binds phosphoinositides. Thus, we also study the role of persulfidation in the ATG18a binding capacity to phospholipids in an in vitro state. We use purified GST18a recombinant protein incubated with feed strips, which are nitrocellulose membranes containing a spot of different phospholipids. And recombinant protein was treated with different concentrations of sulfide and also with a reducing agent. In this case, it was TZ. Spot's intensity was quantified using ImageJ software, and results show that increasing concentrations of sulfide enhance the binding capacity to certain phospholipids, and it was abolished by TZ. So we could say that sulfide significantly activates ATG18a binding capacity to specific phospholipids in a reversible manner. Then we studied the binding affinity of the mutant protein GST-ATG18a C103S, which lacked persulfidation. And results show that mutant protein had a strong, here in this panel, mutant proteins had a strong decrease in its affinity to bind phospholipids, even when it was treated with sulfide, because it couldn't be persulfidated. So we could say that sulfide significantly activates ATG18a binding capacity to specific phospholipids by reversible persulfidation of the specific residue cysteine 103. There's still a lot of work to do, because the next step is to study the effect of persulfidation in vivo in Arabidopsis plants. And to do so, I'm obtaining transgenic plants transformed with YFP ATG18, and also these plants are co-transformed with CFP ATG8 to study the colocalization of this protein ATG18 in the autophagosomes under ER stress, and to study the effect of sulfide in these plants. In parallel, we are also getting the transgenic plants transformed with the YFP fused to the mutant protein, the one, the ATG18, that cannot be persulfidated, and also got transformed with ATG18a to marker, to label the autophagosomes. And of course, we are doing these transformations in a background of a knockout ATG18a to avoid the native protein expression. By the moment we have successfully transformed with these two constructs, so we could check the colocalization of this protein in the autophagosomes, but it's still missing the mutant ATG18a plants, these ones, to compare with the wild-type version of the protein that can be persulfidated. The project right now is kind of stuck due to the pandemic side effects, but as soon as I can go on, I will get more info about this. And finally, I would like to acknowledge all Diane Bassham lab members, but mainly Diane here in the picture, for hosting me in her lab so warmly. And well, of course, a big acknowledge to the institutions, Iowa State University and Universidad de Sevilla. And of course, an acknowledge to funding resources, especially to my grant project under Marie Curie Actions. Well, thank you so much. I will be very happy to answer your questions. Please, if you have some, forward them to me to any of these two emails. Thank you.